If we have connection, Professor Rimmer, the floor is yours.
Thank you very much. Let me just share my screen with you all. Thank you for that very kind introduction. I can confirm that I am not the famous middle distance runner. Can everyone see my screen?
Yes.
Fantastic. So thank you for that very kind introduction. My name is Michael Rimmer. I'm a practicing obstetrician gynecologist based in the United Kingdom, and I'm carrying out some research based at the University of Edinburgh. I'd like to begin by thanking you all for this very kind opportunity to speak to you today about some of the research I'm undertaking. I'd like to speak to you about the novel impacts of extracellular vesicles in the prepubertal testes and the developing somatogonial stem cell niche, and in particular, how treatments with chemotherapy can impact how these spermatogonial stem cells alters their release extracellular vesicles, and some of the exciting data we've identified which may have a meaningful impact on male infertility in later life. I'd like to begin by discussing childhood cancer and male infertility. Sadly, in the United Kingdom, there are 1,800 new cases of childhood cancer each year. But with improving diagnostics and therapeutics, survival is improving year on year. And thankfully, more than 4 out of 5 children diagnosed with cancer today will survive. But this growing population means that there are increasing numbers of survivors with unmet health needs. And one health need in particular is their fertility. And we can see from this graph on the panel, the y-axis shows the cumulative pregnancy index, and the x-axis shows their age. And this graph looks at boys who've had cancer and survived and compares their fertility outcomes, or the numbers of pregnancies they've had, to their brothers who haven't had cancer. And we can see the red line, the survivors of childhood cancer, have a significantly lower number of pregnancies compared to their brothers who haven't had cancer. And this goes all the way up to the age of 45 years. And we can see that this reduction in their fertility potential is a lifelong impact as a result of having cancer treatment as a child. So one thing people always ask us is, can't we just freeze sperm and store it? Well, it's important to remember that we're looking at boys. These are prepubertal boys who do not produce sperm. And sadly, for a young boy diagnosed with cancer today, there are no viable fertility preservation options available for him to use in the future. So how does this damage actually occur? As we can see from this image on the screen, this is a cross-section of an adult testes. We can see the spermatogonium and developing through to mature sperm through the seminiferous tubules. However, the prepubertal testes looks more like this. There's only the spermatogonium and the Sertoli cells. There's no developing sperm. And so we know that chemotherapy, and in particular cisplatin, causes direct damage to the testes, especially the spermatogonial stem cell niche. But what we don't know are the other mechanisms by which chemotherapy can cause this damage too. And it's possible that there are unexplored mechanisms through which chemotherapy can mediate its damaging effects in the testes. And one particular area that my research focuses on is the release of extracellular vesicles. But what are extracellular vesicles? Well, they're small lipid-bound structures released by all cells, and it's a form of cell-to-cell communication. They've got very active cargoes such as RNA, DNA, lipids, and proteins. And importantly, the health and state of the cell releasing the EV, which is often called the parent cell, can change both the number of the vesicles being released and also the composition of these vesicles. Broadly speaking, there are 3 pathways which release these extracellular vesicles. The first, at the top of the screen, is the multivesicular endosomal pathway. These were traditionally called exosomes, or the small vesicles, and these are up to 100 nanometers in size. The second pathway of vesicle release is through microparticles. These bleb off from the cell surface and also contain bioactive cargoes. And the third and largest category of vesicles are called apoptotic bodies. And as a cell undergoes apoptosis and the cell blebs and breaks down, those large blebs are referred to as apoptotic bodies. EVs are extremely diverse in both their size and composition. And if we focus in on an EV, we can see here that there are numerous cargoes which they carry. Importantly, they also carry transmembrane proteins, and therefore they're immunologically inert to the rest of the body. When they're released, they are identified as a normal cell would be. And because of that, they're able to travel large distances in the cell throughout blood, lymphatic fluid, and in fact EVs are found in breast milk, saliva, tears, and cerebral spinal fluid. And so they're able to travel large distances with these cargoes to interact with other cells. And so when they reach another cell, they're able to alter the function of that cell through various mechanisms. But the first way they can interact with the recipient cell is through cell surface receptor binding through a ligand. The second is that they can bind to the cell's membrane and release their cargoes directly into the cell. And the third and fourth options are quite interesting, so they can be completely engulfed by the cell and transported straight to the lysosome, or they can be engulfed and trafficked to a particular subcellular localization. And once these EVs interact with the cell, they can change how that cell functions and behaves. And as we've started to research EVs more and more, we've realised that their functions are quite diverse. These include developing the pre-metastatic niche prior to cancer metastases. We know extracellular vesicles can confer chemotherapy resistance, and they can even trigger decidualization of the endometrium. But what about EVs in the testes, in particular the prepubertal testes? Well, little is known about EVs and EV signaling between developing sperm and Sertoli cells in the seminiferous tubules. And very little is known about EVs from sperm at all. What we do know about extracellular vesicles in the testes actually begins with the epididymis, and there are broadly three main categories of EVs called epididymosomes, prostosomes, and seminal fluid EVs. And although we know little about the prepubertal testes and EVs, we do know that in the adult testes, the release of EVs and their interaction with developing spermatogonial stem cells is that they're essential for the correct maturation of sperm. And we've hypothesized that EVs released by developing spermatogonial stem cells and Sertoli cells in the prepubertal testes, treated with cisplatin, will then impact other cell populations within the testes. So, how do we actually conduct these experiments? Well, there are no accurate human cell lines that we can use for cell culture. So we use a representative mouse model. So we use GC1 cells, which are representative of prepubertal spermatogonial stem cells, and also TM4 cells, which are representative of Sertoli cells. And when we conduct this research, we are guided by the International Society for Extracellular Vesicles, who have produced a minimum standards document for how to conduct EV research. One of the main issues which this tackles is how to produce robust data to actually truly and accurately characterize the extracellular vesicles you've identified, and in particular, how we deal with FBS, or fetal bovine serum. Many of us will culture cells in fetal bovine serum or fetal horse serum. However, a lot of people don't realize that actually FBS contains billions and billions of extracellular vesicles. And so we're able to filter our FBS to remove the extracellular vesicles so that we are confident that the vesicles we collect in our cell culture are released by cells. But how do you actually know if your FBS has the vesicles removed or not? Well, we can use nanoparticle tracking analysis to detect extracellular vesicles suspended in their chamber. So we can see here the extracellular vesicles moving, suspended in liquid, and they move through Brownian motion. We can then shine a laser through the chamber, and that will allow us to visualize the EVs. So if we look at these images here, on the left-hand panel, we can see our negative control. So this contains minimal vesicles. But the second-hand panel, with our normal media, this is what normal media and 10% FBS looks like. And as you can see, it contains a large number of extracellular vesicles, which would be a massive confounding factor in any experiments reporting on vesicles. However, we're able to filter this media to remove the vesicles, and we can see here with our third image, I'll just hover my mouse over it, you can see that this video contains no vesicles. And in our fourth panel over here, this is what our media looks like after 24 hours of being cultured with spermatogonial stem cells. And we can see it contains a large number of extracellular vesicles. But because we've been able to robustly remove the vesicles from our culture serum, we're able to be confident that what we've obtained are actually extracellular vesicles. And so what do our EVs look like? Well, once we shine the laser through using nanoparticle tracking analysis, we're able to both quantify the size of the EVs and the abundance of them. And we can see here that the majority of our spermatogonial stem cell derived EVs from our GC1 cells are less than 200 nanometers in size, and these would be classified as small EVs. So although we're confident that we've generated EVs from our cells, are there any other confirmatory steps that we can do to be sure and confident that what we have identified are actually extracellular vesicles? Well, if we go back to this image we saw previously, we can see some of the common surface markers of EVs, such as CD63, CD81, and CD9. These are widely accepted markers of EVs, and again, agreed upon by the International Society of Extracellular Vesicles. And so we were able to use fluorescent primary antibodies to identify these markers on our vesicles. And this is an important confirmatory step to identify EVs. And so you can see here, using our spermatogonial stem cell derived EVs and D-storm imaging, we can see that we've got 3 small vesicles between 50 to 80 nanometers in size. And we can see here that they are individually CD9, CD63, and CD81 positive. And this just demonstrates that there's both diversity in their size, but also their surface markers. But if I was to show you a larger vesicle, again from a spermatogonial stem cell, this is about 200 nanometers in size, and we can see that this vesicle is positive for all 3 markers. And again, it demonstrates how heterogeneous our population of vesicles are. So how do we actually determine the impact of cisplatin-derived EVs on cisplatin-naive cells? Well, I'm able to culture my spermatogonial stem cells or Sertoli cells in cisplatin, and we can see over time that they release extracellular vesicles into the cell culture media. We're then able to incubate these EVs with our treatment-naive cells, and we're able to also incubate a fluorescent readout of cell activity. And what we look at is the activation of Cleaves caspase, which is a marker for cell death and apoptosis. We are then able to culture our cells in something called an IncuCyte, and this is live cell imaging. This machine sits inside a cell culture incubator, and it's kept at 37 degrees C, and our cells will continue to proliferate and grow. And we can take images over time of the cells in real time. And we can see here from our first image, we can see the outline of our cells in grey, but we can also see lots of green dots. And the green dots is the fluorescent readout of the cleaved caspase, and these are cells which are dying. We can then remove the cells just to identify the caspase detection, and then we can use machine learning to quantify the amount of green we can see. And we can see here we've got the green and a small purple outline, and the machine can effectively go and count the amount of apoptosis it can see in a single image. And this lets us compare EVs derived from cells treated with chemotherapy at different doses. So how do our spermatogonial stem cell EVs impact treatment-naive spermatogonial stem cells? So if we go back up to this image of the testes here at the top, we can see these EVs have come from our spermatogonial stem cells. And looking at the graph, we can see on the y-axis the proportion of apoptosis. And we can see along the x-axis we've got our control here, which we've called vehicle control. And we can see as we move along, we can see different doses of cisplatin-derived EVs from our spermatogonial stem cells. And we looked at 0.5, 10, 100 and 200 micromolar doses of cisplatin. And we can see that there's no significant increase in the proportion of apoptosis seen in our treatment-naive cells. But this led us to think, is this simply a dose-response effect? Have we incubated enough EVs with our treatment-naive cells? So we tried to incubate more EVs with our cells, and so we incubated 2.5 times the number of EVs as seen in the blue panel, and 5 times the number of EVs as seen on the purple panel. And we can see when we increased the number of EVs to cells, there was still no increase in the amount of apoptosis seen in our spermatogonial stem cells. So, what can we conclude so far? Well, we can see no identifiable impact of spermatogonial stem cell derived EVs on treatment-naive spermatogonial stem cells. But what about Sertoli cells? Aren't they important? Don't they form the seminiferous tubules, which are important for developing sperm to travel through? And so we thought, well, what happens when we treat Sertoli cells with cisplatin, and how do those EVs impact other cells in the testes? So we can see here from our next figure, again, if we move up here to look at this image, we can see that EVs derived from our Sertoli cells, then incubated with treatment-naive Sertoli cells, actually do trigger apoptosis. And we can see that there's a significant increase in the proportion of apoptosis detected with EVs released by Sertoli cells treated with 10 micromolar dose of cisplatin. And the ability of Sertoli cell EVs to trigger apoptosis in treatment-naive Sertoli cells has yet to be reported in the literature. And this is a novel finding which you didn't expect to see. And so this made us think, what would happen if we incubated these EVs with our spermatogonial stem cells? Would they trigger apoptosis in those cells? And so we were able to generate EVs from our Sertoli cells and then treat them, incubate them with treatment-naive spermatogonial stem cells. And as we can see from this graph, on the y-axis, we've still got our proportion of apoptosis. And what we don't see is an increase in apoptosis, but quite the opposite, in fact. We can see a background reduction in levels of apoptosis in our 100 micromolar cisplatin dose. Now you might say, well, this is a small effect which you've seen, and our 0.5 and 10 micromolar doses aren't significant, and this may just be an artifact where we've stumbled across significance. But these experiments may require increased N numbers in order to truly determine if this is an accurate result. But what we wanted to do was figure out what actually happens when these EVs interact with the cell. Well, as we can see from a figure which we saw earlier in our presentation, they can interact with the cell surface receptors, they can release their cargoes at the cell surface, or they can be transported within the cell to a subcellular location. So we generated our extracellular vesicles by treating cells with cisplatin, and we were able to fluorescently label their outer membrane with Alexafluor 647. And we can see on the left panel image here, we can see the bright field image of the cell, and we can see in the middle image, we can see our fluorescent labeled extracellular vesicles. And here we're able to superimpose those images, so you can see the vesicles superimposed on the outline of the cell. And this imaging, again, is real-time live cell imaging, and we're able to see the vesicles moving in real time inside the cell. So you can see on the left-hand panel with the purple-stained EVs, you can see them moving throughout the cell cytoplasm. And we're able to track individual EVs moving. So on this middle panel here, where you can see all the green and red lines, these are the individual tracks which the EVs are travelling when they're inside the cell. Tracks which are green or lighter in colour are the slower moving vesicles, whereas those which are red are the faster moving vesicles. And so we're able to actually zoom in on the EVs moving. And so this is the same image, but we've zoomed in. And we can see as the EVs are moving on the left-hand panel, we're able to draw in real time the tracks which they take. And as we can, although it's not stained in this image, the nucleus of our cell is just up here. We can see a differentiation of the nuclear pore just here, the semicircular outline. And we can see here that EVs which are further away from the nucleus are moving faster. Now we don't know what this means. Are they moving faster because they're trying to get to the nucleus? As we can see from our purple image, there's a lot of aggregation of EVs around the nucleus. Or, although if you look at the images, some of them are travelling away from the nucleus. So we still need to determine what the meaning of the speed of EVs is. But it led us to think, is there a difference between cisplatin-derived EVs and our control EVs and how they behave inside the cell? So first of all, we wanted to look at the amount of EV uptake by treatment-naive cells. So we can see here from this figure, when we fluorescently labelled our extracellular vesicles, we can see that with our 0.5 dose of cisplatin EVs, there was no significant increase in uptake. But with our 10 micromolar dose, we can see that there was a significant increase in EV uptake by treatment-naive cells. Now this is important because remember, EVs are a way that cells talk to other cells. So not only have we seen that they mediate phenotypic changes in cells, but we can also see that more cisplatin-derived EVs are taken up by treatment-naive cells. It's almost as if the cell releasing them is trying to tell other cells they're being treated with cisplatin. But what about the speed of EVs inside treatment-naive cells? Well, we can see from this graph here, we can see on the y-axis, we've got the pulse per frequency, so the number of vesicles we detected. And we can see in the light blue graph, these are our cisplatin-derived EVs. We compare that to the purple lines, which are control EVs. And there are two things to take away from this graph. The first is that there appear to be more EVs taken up again, by more cisplatin-derived EVs taken up compared to control EVs. But that if you look at the overall shape of the graph, the slope, that they're almost the same. And so even though more EVs are taken up when they're released by cisplatin-treated cells, when they get inside the cell, they appear to move at the same speed. And this data is, in terms of looking at their speed, we could identify no significant difference. So what experiments do we plan to do next? Well, we'd like to examine the cargoes of these vesicles and see what's inside them. What's causing these phenotypic changes in recipient cells? So we'd like to carry out both transcriptomics of the EVs and proteomics. The reason we want to do transcriptomics is because we know that the siRNA inside EVs is a known feature of EVs which can mediate changes in recipient cells. We'd also like to understand, do our TM4 EVs compare chemotherapy resistance to our spermatogonial stem cells? So the next set of experiments we're going to do in the lab is we're going to culture our spermatogonial stem cells, treat them with Sertoli cells, and then incubate them with chemotherapy. And will we see less death in our EV-treated cells compared to controls? The ability of EVs to compare chemotherapy resistance is well characterized, and this may be an additional feature of our Sertoli cell EVs in the testes. We'd also then like to examine the proteomic changes in these recipient cells to understand what are the real changes that occur inside these cells when they take up these EVs. And then finally, we'd like to investigate how EVs impact the relevant human tissue by incubating our EVs with prepubertal testes tissue and examining these tissues for cell death. So what can we conclude so far? Well, we've identified that cells treated with cisplatin release more EVs than control cells. These EVs then interact with cells and are incorporated in the cell more readily. And we've seen that EV-derived, cisplatin-derived EVs more readily taken up compared to controls. Our Sertoli cell cisplatin-derived EVs can induce apoptosis in treatment-naive Sertoli cells, but also seem to induce lower levels of background apoptosis in our spermatogonial stem cells. We may have identified a novel mechanism by which chemotherapy can impact the testes, and in particular, how Sertoli cell EVs trigger apoptosis in other Sertoli cells may make a meaningful contribution to male infertility in later life. And it's by understanding this mechanism through which these EVs confer cell death, which will allow us to potentially develop novel strategies to try and reduce the impact of chemotherapy treatments in boys with cancer. I'd like to thank you for your time and attention today and for your kind invitation to speak. I'd like to thank the supervisors of this work, Professor Rob Mitchell and Professor Chris Gregory, and I'm very happy to take any questions. Thank you.
It's interesting where we talk about treating oncology treatment in the young people, and also how to preserve fertility in the later age. These small particles may play a critical role in cell-to-cell interaction, and it's very interesting talk. And also thank you for the beautiful picture we see in your presentation, colorful pictures. Now the topic is open for discussion. Here from the hall, you may ask a question to Professor Rimmer. Also, our participation online. If you want to ask a question or make comment, the floor is yours. Here from the hall, we have comment from Professor Kyrkchiev.
hear your talk. And I would like to ask you the following. If the EVs you are dealing with are induced, have you any means to compare the cargo of induced? No? Well, I have to start again. My question is, if the EVs you are dealing with, they are induced by treatment with cisplatin, and do you think there is a difference between the cargo of the EVs when comparing the normal, non-induced EVs and the induced EVs? And a more general question I have, are there any data about any way to control the cargo of the exosomes of EVs in general, compared to the fact that in vitro, when cells are treated, let's say in hypoxemic conditions, it's possible to increase content in the exosome. If that is possible to control in vivo the cargo of exosomes, that might be a new treatment or a new approaches for developing a new novel, as you said, strategy for treatment. Thank you, and thank you once again for your lecture.
Thank you for your question. I didn't quite catch all of that, but I think you asked, is there a difference between our chemotherapy-induced EVs and our control EVs? With our chemotherapy-induced EVs, we can see that more are being produced by the cells and that those EVs are more readily taken up by recipient cells. And we think this is quite important because we know cells release EVs as an active way to communicate with other cells. So we don't think that's a mistake that they were releasing more. We think they're trying to communicate with other cells more. I think you asked, can we confirm this in vivo as well? In vitro, the microphone cut out slightly. What we're doing to see do these have impact on human tissue is we're able to generate our vesicles in large numbers using a cell culture model, and then we can concentrate those down and incubate those vesicles with human prepubertal tissue. We're then going to undertake two experiments. The first is we're going to try and identify the vesicles in the tissues. Do our vesicles get taken up by certain cell populations preferentially in tissue, or do they trigger the cell death in cell populations? If we want to start Tory cells in the cell culture model, we can trigger apoptosis Tory cells. When we do the we still trigger apoptosis when incubated with tissue. I think that was your question. The microphone cut out there, sorry.
Professor Riemann, can you check your microphone, please? Because we have a noise here in the hall.
I can hear you. Can you hear me okay?
Okay, yes. Now it's clear. Thank you.
Did you hear my response?
Professor River, could you hear us?
I can hear you. Can you hear me?
Yeah, we can hear you now, but we didn't hear the last 2 minutes from your talk. Could you repeat it?
Yes, absolutely. So I think the question was, how are we going to examine the impact of these EVs in the human tissue? And so we're able to generate the EVs in large numbers from our cell lines and concentrate these EVs. We're then able to incubate these EVs with prepubertal tissue. So a boy diagnosed with cancer today will have a small amount of testes tissue removed before treatment, and a small part of that will come to the lab for research. And so we're going to incubate the extracellular vesicles with the tissue, and we're going to do two things with that tissue. The first is that we're going to examine that tissue for EV uptake. Can we identify that our control or cisplatin-derived EVs are preferentially taken up by different cells in the tissue. And the second thing we're going to do is we're going to examine that tissue for cell death. So do our Sertoli cell EVs, which appear to be triggering apoptosis in our cell line models, still trigger apoptosis in the tissue? Or do they trigger apoptosis in other cell lines which we haven't looked at? I think that was your question. I couldn't quite hear you at the start, sorry.
Thank you for your answer, but my question really is it possible to control the cargo, the composition of the extracellular vesicles in vivo? Are there any ways to induce not only their quantity to increase their amount produced by cells, but to control their their cargo, the proteins, the mRNA, and everything that is inside the cells, the vesicles?
Thank you. So that's a really great question, actually. In terms of their EV release, in a cell culture line, you can try and use, there are certain drugs you can use, one of which is simvastatin, to inhibit EV uptake. But in terms of EV release and cargo loading, we don't fully understand all the mechanisms by which cargoes are loaded into EVs. So that would be great if we could identify particular cargoes in our EVs which are triggering apoptosis. We could stop them being released. But no, we're not able to influence how these cargoes are loaded yet. What we can do, however, and what is being explored, is preferentially putting cargoes into the EVs. So there's some research at the moment which is using EVs as novel vectors to deliver siRNAs. So although we can't control what goes in them, if we can take them out of the body, we can artificially load other things into the EVs and then replace the EVs and use that to deliver a drug. So it's not quite what you asked, but it's fairly close.
Well, thank you. Maybe we need some more time to discuss it personally, and I will invite you to come to Bulgaria next May when we have our National Congress. Then we can discuss it in for much longer time. Thank you very much.
Thank you.
Enjoy your day.
Thank you, Professor Riemann, again. You may continue listening our next topic.
Okay, if there are no any questions. Знаешь, что телевизит.